Welcome to the deep dive. We've got a lot of questions from you about some core brain stuff, specifically receptors.
Ionotropic versus metabotropic, right? It's a fundamental difference.
Exactly. So today, our mission is to really unpack that. What sets them apart? Why does it matter for, how our brains work? And even how drugs work, too.
Right. And looking at the notes here, the absolute core distinction, the main thing, is how they react when a neurotransmitter shows up.
Okay.
Ionotropic ones are, well, they are the ion channels themselves. Think of it like a gate that unlocks instantly. Neurotransmitter binds, pop, the channel opens, ions flow.
Direct, like a key in a lock opening a door straight away.
Precisely. But metabotropic receptors, they're different. They aren't channels. They sit next to the channel, or maybe not even that close.
So they don't let things through directly.
No, they have to kick off a process inside the cell first. It's more indirect.
Okay, so direct versus indirect. That must mean a difference in speed, right?
Yeah.
Ionotropic sounds fast.
Very fast. That direct opening means the signal transmission between neurons, the synaptic transmission, happens almost instantaneously. Quick on, quick off, usually.
And the metabotropic ones?
Slower. Yeah, definitely slower. Because they have to activate things inside the cell first, there's a delay. But their effects can be much more widespread and longer lasting.
Interesting. Can you give us some quick examples
Yeah.
of the fast ones, the ionotropic?
Sure. A classic one is the nicotinic acetylcholine receptor, crucial for muscles, also in the brain. And GABA A receptors, they're a big target for anti-anxiety meds. They're ionotropic too.
GABA A, right, for calming things down quickly.
Exactly.
So how does the slower indirect system work then, the metabotropic one? What happens inside the cell?
Often they work through what are called G proteins. Think of them as like internal messengers. The receptor gets the signal outside, then activates a G protein inside.
Oh yeah, middleman.
Sort of, yeah. And that G protein then goes on to do other things. Maybe it triggers the production of other molecules called second messengers.
Second message.
Right. And those can then affect various things. Maybe they open or close ion channels indirectly, or change how genes are expressed, or activate enzymes. It's like a cascade.
Oh, okay, a whole chain reaction inside the cell.
Exactly. Which takes time, but allows for much more complex sort of modulation of what the cell is doing, not just a quick on-off switch.
So the receptor type really dictates the whole character of the signal, not just if it happens, but how and for how long.
Absolutely. Ionotropic gives you those rapid, short changes in the cell's electrical state, essential for, quick reflexes, processing sensory info fast. Metabotropic allows for slower, more sustained changes, fine-tuning.
Which sounds like what you need for things like learning, memory, maybe mood. Stuff that isn't just an instant reaction.
Precisely. Those kinds of brain processes rely heavily on that metabotropic signaling because you need those lasting modifications, that plasticity.
And this difference, direct versus indirect, fast versus slow, that must be huge for developing medicines.
Oh, absolutely huge. You can design drugs to selectively target one type or the other. So like we said, GABA enhancers for quick anxiety relief, that's ionotropic.
Right.
But then drugs for, say, Parkinson's might target metabotropic dopamine receptors, aiming for a more sustained modulatory effect on movement control circuits.
Makes sense. And what happens if this signaling gets messed up, if either system goes wrong?
Well, disruptions are linked to quite a few neurological and psychiatric disorders. Problems with GABA signaling, for instance, can be involved in anxiety, insomnia, epilepsy,
Okay.
and issues with, say, metabotropic glutamate or dopamine receptors are implicated in things like schizophrenia, depression, Parkinson's. It's all about balance.
How do scientists actually study these tiny things?
Various ways. Electrophysiology is a big one, especially for ionotropic receptors. It lets you measure those rapid electrical currents directly. And then there's molecular biology, genetics, figuring out the structure, how they bind, how they're built.
Got it. And maybe one more therapeutic example. How else are they modulated?
Sure. So besides boosting GABA, sometimes you want to block receptors. There's research into blocking certain metabotropic glutamate receptors, for example, in conditions like Fragile X syndrome, trying to sort of rebalance brain circuits that have become overactive.
Okay, fascinating. So let's try and sum this up. The fundamental split is ionotropic receptors are fast ion channels opened directly by neurotransmitters.
Right. Quick, direct action.
Whereas metabotropic receptors use internal messengers, like G proteins, to trigger slower, more complex, and longer-lasting effects inside the cell.
Exactly. Indirect modulatory.
And this difference impacts everything from signal speed to learning, mood, and crucially, how we design drugs to treat brain disorders.
You've got it. It's really about having these two different toolkits for communication in the brain, one for speed, one for finesse and lasting change.
So here's something to think about. How does this constant interplay, this balance between the superfast signals and the slower, more shaping signals, contribute to the richness of your own thoughts, your feelings, your behaviors?
Yeah.
What might happen if that balance tipped too far, one way or the other?
Interesting question.
Definitely something to ponder. Thanks for joining us for this deep dive.